Swayam Prabha. Digital India. Educated India.
Welcome to my course on genome editing and engineering. In this module, we are going to discuss about the CRISPR-Cas9 technology. As many of you may be knowing, CRISPR stands for clustered regularly interspaced short palindromic repeats. In this lecture 1, we are going to discuss about the basic biology of CRISPR system in bacteria. Let's start with the work of Ishino et al., who in 1987 was working on an interesting enzyme called alkaline phosphatase, which has many isozyme forms. They were looking into the mechanism of conversion of the various isozyme forms in E. coli, and while doing so, they discovered a series of 5 repeated palindromic nucleotide sequences in the 3' end flanking region downstream of the IAP gene. The repeat sequences were separated by variable sequences of uniform length with no homology to each other. Although of no relevance to their work, they nevertheless dedicatedly reported about this unusual sequence structure with an unknown biological function in their concluding part of their manuscript. Thereupon it was soon forgotten by the scientific community. In 1992, another researcher called Francisco Mojica, working at the University of Alicante in Spain, was reviewing the genome sequence data from an extremely halophilic microbe called Halopharax mediterranei, and he noticed that in this particular organism there were 14 unusual DNA sequences, each of which was 30 bases long, and each of these sequences read roughly the same backwards and forwards, and they repeated every 35 bases or so. Mojica developed a lot of interest in these sequences. However, he had to struggle to find funds to pursue research on this new discovery, and he was even ridiculed by peers and decision makers for his interest in these strange little repeats. There are many repeats in many organisms. We have known them for years and still don't know how many of them work, many of his peers and criticizers would say, and they would discourage him by telling, don't care about repeats so much. At the time, no one, including Mojica, knew that these repeats were going to change the course of biotechnology revolution in the coming decades. The term clustered regularly interspaced palindromic repeats, CRISPR, was coined by Mojica in correspondence with a team of Dutch scientists led by Ruud Janssen at the Utrecht University in the year 2002. Janssen in the same year identified another set of sequence present with CRISPR, which he named as CRISPR associated genes or Cas genes. By 2005, Alexander Boltin found that spacer sequence between the CRISPR sequences shared similarities with the DNA of viruses. They also discovered 2 to 6 base pair long protospacer adjustment motif or PAM, a conserved sequence present at the end of sequence similar to spacers in the target. It was hypothesized that CRISPR has some role in immunity of host bacteria, although it was not understood exactly how. Let us now look into some of the important discoveries in the field of CRISPR. As we have already discussed about the work of Ishino et al., in 1987, they discovered some tandem repeats sequences downstream of the IAP gene in E. coli while looking for the mechanism of isozyme conversion of alkaline phosphatase. Then we know about the work of Mojica, and it was in 2002, which we have already discussed, that he along with Mojica along with Janssen gave the name CRISPR. In 2005, we know that the spatial sequences in bacteria and archaea were found to be highly homologous with their viruses which attacks them. By 2012, this basic knowledge was soon converted into a technology. It was possible to do in vitro genome editing with CRISPR-Cas9, and this was developed in 2012. The very next year, this technology was used for modifying eukaryotic cells by using this CRISPR-Cas9 technology, and in 2014 there was discovery of the precise method to modify genes in eukaryotes with this CRISPR-Cas9. In 2016, oncogene mutations were inactivated by using CRISPR-Cas9 technology, and 2017 CRISPR-Cas9 was used to modify the beta globin gene in human embryos. In between these discoveries, there are many important discoveries which we are not discussing in detail. Kindly refer to available literature if you are interested. 2019 is considered a black year in a way. He Jingjiangkui in China was jailed for deliberately violating medical regulations and for rashly applying gene editing technology to human assisted reproductive medicine. In 2020, amidst the corona pandemic, Emmanuel Charpentier and Jennifer A. Doudna was awarded Nobel Prize for the development of a method for genome editing, which was basically the CRISPR-Cas9 technology. So from 1987 up to 2020, you can see the trajectory through which this technology and discovery moved through, right up to the year of 2020 when it was honored with the highest award. The developments has not stopped. Many new developments are coming in of late. We will discuss some of those in this particular course and these few lectures. Let us go to the year 2007 where when Barango et al. Found out that these sequences, which were once ignored for their unknown biological functions and was being ridiculed by peers as mere repeats and now named as CRISPR, together with associated Cas genes, form an adaptive immunity which provides resistance against bacteriophage infection. Basically, the immune system provides these kind of defense in higher organisms which are multicellular, but finding such a corresponding system in a very small single cellular bacteria was quite astonishing. This was published in Science. As you can see, this paper, CRISPR provides acquired resistance against viruses in prokaryotes and is considered a landmark paper. Let us now go to a concept called Markov chains. Many of you may be already knowing. This was introduced by Andrew Markov in 1906 and is used to model a discrete time, discrete space stochastic process of various domains like finance, engineering, physics, and even genetics. The Markov chain can be described as a triple of S, X and P. A set of states is denoted by S with X random variables and a transition probability matrix of P. In simple terms, a Markov chain is a sequence of random variables within a finite state space with values in S for which the transitional probability P of the state at the time t is given by the transitional form, the state and the time t minus 1 with probability p, which is called as the Markov assumption. This time t minus 1 is important for the next event. We will discuss about that how. The random variables transition from one state to the other based on an important mathematical property called Markov property. Discrete time Markov property states that the calculated probability of a random process transitioning to the next possible state is only dependent on the current state and the time, and it is independent of the series of states that preceded it. To make it simple, right from your kindergarten days to middle school to high school, you have various levels of performances, or you acquired knowledge step by step, right from learning the alphabets. So finally, when you graduate out into college, what is important is your final score in the 10th or 10 plus 2. Your score at 10th leads you to the 10 plus 2, where you decide whether to go to the art streams or the commerce streams or the science streams. And supposingly you have taken up the science stream, again your performance at the 10 plus 2 would decide whether you are going to management, science or engineering or some other discipline. And this decision is not actually having any impact from your earlier results prior to 10 plus 2. Okay. So now we are going to use this example to understand some of the important concepts in the CRISPR biology. So genetic adaptive mechanism of CRISPR-Cas follows a Markov chain model. So here you can see there is a initial unprotected state where you have a virus which is not immune to you have a bacteria which is not immune to certain viruses. But something happens in the due course of time, the bacteria acquires some capability and it lends up or goes through an intermediate phase, and due to which it acquires immunity. So from a unprotected state here, the bacteria gains a protected state. And this leads to the final bacterium state. Now, if you look into each state, there are two probabilities. This is a bacteria which is unprotected. This bacteria will undergo reproduction and survive because it is not attacked by any virus, but its other partners or other daughter cells were attacked by the bacteria and were killed, attacked by the virus and were killed. In the meantime, the bacteria acquires some weapons and it is in intermediate state. This also again would reproduce, and this is not being attacked by any virus, so it survives, but some of its other daughter cells are attacked by viruses and these are killed. Soon this acquisition of the weapon becomes permanent and the bacteria again reproduces, and this is not yet attacked by the virus, so it survives while a population of it is killed. Okay. So which means the immunity, although it has been in the stage of acquisition, is not yet getting expressed properly and the organism is still being killed, but after this stage it enters a final stage where the immunity against the invading virus becomes permanent, and in this case the bacterium kills the phage. So you can see from this stage to this stage the scenario is totally altered. Now this bacterium final stage is dependent on the immediate state, which is the T minus 1, and it is although dependent on this stage and this stage, but it is the events in this stage which decides the final outcome, and this is what we call as a Markov chain model. So, basically the transition events for the state change on individual bacterial or archaeal cell can be described and depicted as shown in this figure, which we have discussed in detail. Each event in this Markov process occurs with a probability proportional to the event's rate called phi I. So, this is phi 1, phi 2, phi 3, and the other states phi 4 and phi 5, each state depicting one kind of event. Phi 4 is the event where the bacteria reproduces and survives. Phi 5 is the event in which the bacterium reproduces, but it is killed by the phage. And phi 3 is the stage where the bacterium kills the phage, which is altogether the opposite. In the case where a cell begins in an initial state without protection against a particular phage, it must obtain a spacer and express it as a cRNA, which we actually call as a CRISPR RNA. So, these are the things that we are going to discuss, how this acquisition is being done. If this particular phage strain attacks again, the bacterium uses cRNA to interfere. At its state, there is a probability that the bacterium will reproduce or be killed by a phage. This chain of events could be broken down further to include the probability of bacterium phage interaction and the probabilities of a lytic or lysogenic phage attack. Tau I are the assumed characteristics time scales of each stage of immunity. So, we have here tau 1, tau 2 and tau 3, and you can remember this, which we are going to use in the future discussions. So, the genetic adaptive mechanisms of CRISPR-Cas9, CRISPR-Cas follows the Markov chain, what we have discussed, and we also discussed about these tau 1, tau 2 and tau 3. Now, if you look into the figure on the right side, this corresponds to the stage called adaptation, this corresponds to the stage called expression, and this corresponds to the stage called interference. So, we are now trying to describe this Markov chain model in the physical terms, what goes on in the bacteria's adaptability against a viral attack. So, you can see here a virus attacking the bacterial cell, and it is pushing inside the genetic material. Something called spacer acquisition from this target is going on. So, this DNA is being cut and incorporated into somewhere. That place is actually basically the CRISPR-Cas loci, and then these may have been actually attacked by another virus in the past, and that genetic information was also included here, and now this is part of the CRISPR-Cas loci. So, the bacteria keeps on accumulating all the information DNA fragments from viruses which attacked it in the past. Now, we know that bacteria are very short-lived organisms, and they divide. E. coli divides as fast as in 20 to 22 minutes. So, these sequences which are acquired in one single generation would actually be passed on to the next generation, and that next generation would add up the new information resulting out of the viral attack. So, we know from this small discussion that this is also a heritable character. So, we are going to discuss about that again in the future. Now, there is a stage called expression where these loci is transcribed and to produce CRISPR RNA, and then these CRISPR, and this is called the expression stage and or the intermediate stage, and then finally these will lead to the interference or the where the bacterium will kill the virus. So, the CRISPR locus is now little bit familiar to you. This is present in about 84% of archaea and 45% of bacteria according to CRISPR DB. CRISPR-Cas locus functions as a adaptive and heritable immune system of prokaryotes against invading viruses. So, in our case, whenever we take a vaccine that lasts our lifetime, and then if an individual is vaccinated in his childhood that immunity can go up to adolescence, and with booster doses it may be going a little bit further. But in the case of CRISPR-Cas immunity in bacteria, it actually passes on from generation to generation, because here the immunity is stored in the form of information in its chromosome, while the immunity that we acquire by say vaccination do not go to the next generation. However, of course, in certain cases there is mother to child transmission of immunity, but it will not happen in the male population. So, upon infection with virus, the viral DNA is acquired and integrated into the CRISPR sequence, as we have shown in the figure earlier. Future infections would lead to targeted cleavage of the invading viral DNA, having similarity to integrated spacer sequence of CRISPR. Now, you can see this in simple terms, the CRISPR-Cas loci, where you have the Cas genes and you have the CRISPR array. Now, these are all interspaced by phase derived spaces, the colored angles, and these are the dark ones are the repeats. Let us see a little bit closer how this system works. So, once a virus attacks a bacterial cell and it injects the DNA inside the bacterial cell, there is a system inside the bacteria, the CRISPR-Cas loci, as we already know. They produces a complex called Cas1 Cas2 complex. This Cas1 Cas2 complex will go and cleave a selected portion of these viral DNA and insert it into the existing CRISPR array. So, with each infection, the length of this array keeps on increasing. So, this CRISPR array is also kind of a scoreboard or scorecard, where we can know how many times this particular bacterial strain has been infected by virus in the past. The CRISPR-Cas system can be categorized into two classes based on the structure and function of Cas protein, which are further subdivided into 6 types. So, class 1 includes type 1, 3 and 4, class 2 includes type 2, 5 and 6. Type 1, 2 and 5 systems recognize and cleave DNA. Type 4 can edit RNA, and type 3 edits both RNA and DNA. As we have already shown that Cas1 Cas2 system selects the fragment of DNA to be incorporated into the CRISPR-Cas loci, and today we know that all known CRISPR-Cas systems contain these two proteins, Cas1 and Cas2. The 3 different types 1, 2 and 3 are each characterized by unique Cas proteins involved in maturation of the CRISPR RNAs, targeting of foreign nucleic acid and nucleic acid cleavage. The type 2 CRISPR-Cas system is defined by the presence of a large 1000 to 1600 amino acid endonuclease called as Cas9. Let us look into the role of Cas1 and Cas2 in protospacer DNA acquisition by the host CRISPR array for the CRISPR-Cas adaptive immunity. So, acquisition of the enemy DNA, which is basically the viral DNA, happens in 4 steps. The first step is the protospacer binding and selection. Second step is the 3' overhang cleavage, as you can see here in this figure. The third is the integration, and the fourth is the DNA synthesis and repair. In the integration step, PAM-C sequences in C' OH, the cleavage at site 1 of Cas1 Cas2 is integrated into the spacer side of repeat 1 in the CRISPR array, likely via the second nucleophilic attack, whereas the non-PAM-C cleavage at site 2 is integrated into the leader side of repeat 1, likely via the first nucleophilic attack. Let us look into the crystal structure of these Cas1, Cas2 bound to dual forked protospacer DNA from E. coli. So, you can see here Cas1A, Cas1A prime, this is Cas1A and this is Cas1A prime. Similarly, you have Cas2 and Cas2 prime. Then you have Cas1B and Cas1B prime, and they are shown in their respective colors to make them distinct from each other. The two active sites 1 and 2 are shown with black circles. You can see here a black circle, maybe not visible clearly, but within this circle if you look into, you can see a black circle over here which has become red now due to my coloring. So, these are the active sites 1 and 2. In a crystal structure, both sites are bound with PAM-C CTT. The PAM-C recognition via hydrogen bonding and amino acid side chain stacking in the binding pocket are indicated in black and gray dotted lines respectively. Here you can see the hydrogen bonding and the side chain stacking. Type 2 CRISPR-Cas system are further characterized by the requirement for a unique accessory RNA, the trans-activating CRISPR RNA or the tracer RNA, as well as RNase tree for maturation of CRISPR RNAs. Cas9 is involved as a scaffold for maturation of CRISPR RNAs and is required for cleavage of the double-stranded DNA target. So, you can see here the various genes in these particular loci. Let us look into the class 1 and class 2 CRISPR system a little bit closely. What are the key features and the modularity of their organization? So, you can see here in figure A the general architectures of class 1 which is having multi-protein effector complexes, and class 2 which is having single protein effector complexes. Here the genes are shown as arrows, homologous genes are shown by the same color, genes names follow the current nomenclature and classification, and here are the CRISPR repeats and here are the spacers. So, spacers and repeats you already know in detail, and these are the associated genes, CRISPR associated genes, and you can see here in class 1, Cas1 and Cas2 are present in both, in fact in all CRISPR-Cas system as we have already discussed, but you will see that besides these two highly conserved genes in all the systems, there is variation in the other Cas genes. As already told to you, class 2 has only a single effector protein which may be Cas9, Cas12 or Cas13, whereas in type 2 you have so many Cas3, Cas8, 10, 8 or 10, 11, 7, 5, 6 and so on. So, this is very important for our future understanding of the CRISPR-Cas system in bacteria. So, what are the principal building blocks of CRISPR-Cas system which constitute the diverse types? So, here you will see the class 1 which we described earlier and class 2 and the types under these classes respective classes, and you can see here the various modules used for adaptation and expression and interference, and also some modules associated with the signal transduction, and others are having certain ancillary sequences which may having some helping role or some kind of unknown roles. So, we already spoke about these. For example, this is having type 2 has having Cas9 and then 5 is having Cas12 and Cas13, and in all of these you can see Cas1 and Cas2 are invariably present. In in type 2 CRISPR-Cas9 system, you also have a protein called RNase3, and then you have CSN2. So, we have to remember these proteins in future while discussing the CRISPR-Cas9 system. So, this is a tabulated form about the differences between the various types of CRISPR-Cas system, and you can see here the type and you can see here the signature protein against each type. You have type 2 Cas9, very famous protein, and then in type 3 you have CSM, CMR. In type 1 you have cascade. We will have a little bit of discussion about what is cascade. Then you have the various cleavage products. Here you have single strand SSBs and you have DSBs and so on. Now, let us look into the expression of these CRISPR-Cas loci. Now, let us look into the expression of the CRISPR-Cas loci. So, we know about these CRISPR-Cas loci now. So, these loci or genes get transcribed into a pre-CRISPR RNA. So, here you have all the repeats and all the spacers expressed as a single transcript, and you have to remember that these are basically the sequences of the viruses which attack these bacterial strain in the historical past. Also, they are basically antecedent DNA. So, once these transcription happens and the formation of the pre-CRISPR RNA happens. Now, depending on the class of the CRISPR-Cas system which we just discussed prior to this slide and the type, the processing would be different. Okay, so, this is the diversity and variation in this processing. So, you can see here in under class 1 we have type 1 and type 3, under class 2 type 2 and type 4. So, these pre-CRISPR RNA is handled in different ways by different proteins in these different class and different types. So, for example, in this class 1 type you have the role of Cas6 or Cas5D or cascade here. Here Cas6 plays a role and it involves the CSM or CMR complex, and in type 2 you have class 2 type 2 you have tracer RNA, RNase3 and Cas9, and here you have the CPF1, and then the interference mechanism will also be different. We will be discussing some of these in detail in the next slides. So, as already told to you here in this description of the figure, this pre-CRISPR RNA is further processed by Cas6 in type 1 and 3 systems. In type 2 CRISPR-Cas system, CRISPR RNA maturation requires this tracer RNA, RNase3 and Cas9, whereas in type 5A system CPF is alone enough for CRISPR RNA maturation. In the interference state of type 1 systems, cascade, which is basically CRISPR associated complex for antiviral defense, is guided by CRISPR RNA to bind the foreign DNA in a sequence specific manner and subsequently recruits Cas3 that degrades the displaced strand through its 3' to 5' exonucleotic activity. So, here the type 3A and type 3B CRISPR-Cas system employs CSM and CMR complexes respectively for cleavage of DNA and its transcripts here shown by the black and red triangles, and class 2 a ribonucleoprotein complex consisting of Cas9 and a tracer RNA, CRISPR RNA duplex targets the cleavage invading DNA in type 2 CRISPR-Cas system, and while in 5 the CRISPR RNA guided effector protein CPF1 is responsible for target degradation, and the red triangles here represents the cleavage sites of the interference machinery. So, you can see these sites are all in different locations depending on the class and type. Type 1 and type 2 systems use sort 3 to 7 base pair protospacer adjacent motifs or PAMs for identification of suitable protospacer for acquisition. During naive adaptation by type 2 systems, PAM is recognized by Cas9, while in type 1E systems Cas1 and Cas2 are sufficient for recognition of PAM. Following the protospacer selection, the acquisition machinery performs site specific integration of the new spacer into the CRISPR array at the leader end concurrent with duplication of the first repeat. It has been shown that both the leader sequence and the first repeat are essential for this process. Studies of the E. coli type 1E and S. thermophilus type 2A systems reveal that the leader repeat boundary serves as an anchor for spacer integration. We will be discussing this soon. Now, let us look into the arrangement of the various components in the CRISPR-Cas immunity system in type 2A CRISPR-Cas immunity system in particular. So, you have a CRISPR array over here, and then this is the existing CRISPR array. Okay. So, this is basically the tau 1 state, or yes, and the initial state. Okay. Now, if the virus is attacked by if the bacteria is attacked by a new virus, in the presence of Cas1, Cas2, Cas9, CSN2, a new spacer is acquired. Okay. So, these viral DNA having this particular color code is added here. So, this is the tau 3 state. So, here you can see this up to the yellow, this is up to this point it is same. So, new one is added over here. So, this is just schematics, it does not happen that straight away, we will discuss how it exactly happens. This is just to make the simple conceptual presentation that the viral DNA is added to the existing CRISPR array with the help of the Cas1, Cas2, Cas9 and CSN2 proteins. And these Cas1, Cas2, CSN2 DNA spacer capture complex. So, these complex will, leaving aside the Cas9, these complex will bind to this viral DNA. So, you can see here this is the viral DNA having the similar color code, and these are the 4 Cas1 dimers, and then you have 2 Cas2 dimers, and you have 2 CSN2 tetramers. So, this is a 90-degree rotation and a different view of this same structure, where these Cas1, Cas2, CSN2 forms a complex with the viral DNA to cleave it and to cut it from the viral DNA and paste it into the CRISPR array, existing CRISPR array. So, you can see here roughly around 30 base pair of dsDNA. This is protected within this complex, and this will be pasted into the existing CRISPR array. And if you remember the work of Isino and Mojica, and they spoke about something around 35 base pair length spacers or repeats. Okay. So, in addition to the Cas1, Cas2, Cas9 type 2A CRISPR-Cas system also include this CSN2, which is considered a subtype specific signature Cas protein. CSN2 has been implicated in the adaptation stage of CRISPR mediated immunity, since was shown to be required for the acquisition of new species during in vivo adaptation experiments. When as pyrogenes type 2A Cas operand was expressed in E. coli, 4 Cas proteins were co-purified, suggesting the formation of a multi-protein Cas complexes. So, these are the experimental evidences which led to the suggestion of these kind of structures occurring. How does the interaction between Cas1, Cas2 and CSN2 proteins occur exactly within this complex? So, we spoke about these 4 Cas1, 2 into Cas2, 2 into CSN2, and you can see here this is a Cas1 dimer here, this is a Cas again here, CSN2 head, and this is a Cas2 dimer over here, and this is as the 90-degree rotation to show the other side of this particular complex, and these are in more details the structure of these particular complex. So, this is a more detailed view of the top of the complex here, right. So, in the Cas1 dimer interacts specifically with just the CSN2B promoter of each CSN2 head domain. This model is shown as a cartoon with the repetitive interacting chains outlined by a transparent molecular surface filtered to 20 angstrom resolution. Now, this is an overlay of the EF Cas1 dimer and Cas2 terminus from the spacer bound crystal structure with a Cas1 dimer from the monomer complex. Both the asymmetry of the Cas1C terminal domains and the interaction between the C terminal strand of Cas2 and the Cas1B and terminal domains are conserved over here. This is a close up of the Cas1 CSN2 interface. Different residues on a single CSN2 protein interact with the symmetrical surfaces of the Cas1 and terminal domains. Now, this is a proposed scheme for spacer acquisitions. Now, we know that these Cas1, Cas2 CSN complex binds to the viral DNA, but how do these acquisition really takes place? So, this is a proposed scheme as presented over here. So, you have this viral DNA fragment which has a palm site, okay, which precedes the potential protospacer. Now, these Cas1, Cas2 CSN complex engages on the free DNA and sites, and while the Cas9 CR tracer RNA would bind to the palm site. So, CSN2 protein has high DNA affinity, it binds to the DNA ends, as a result of which this Cas1, Cas2 CSN complex would engage free DNA ends in the cells and encircle DNA within the complex. This Cas CSN complex would then slide on the DNA upon binding. It goes towards the Cas9 complex and it will keep on moving until it encounters a Cas9 protein that is bound to a palm sequence. At that point, the DNA could be cleaved by an as yet identified cellular nucleus, releasing Cas9 and the Cas1 2 CSN complex, encapsulating a 30 base pair DNA fragment, as you can see in the figure, and as a new spacer ready for integration. So, this cleaving is or picking up of the viral DNA is complete at this stage. Integration of the spacer would require dissociation of the complex to release the Cas1 Cas2 complex in the conformation compatible with spacer integration. The unknown the process is much more complicated than as currently understood, and work is going on to elucidate the details of how DNA is captured in these structures and how it is incorporated into the CRISPR arrays. A lot of details already known, but certain information are missing. So, this it still remains many of these things still remains unknown in certain aspects. So, let us look into the structure of the Cas9 protein. For example, we are looking into the structure of the Streptococcus pyrogenes, and these contains about 7 domains with some basically are subdomains. So, you have these REC1, REC2 and REC3. Then you have a bridge over here, which is a bridge helix. Then you have RAVC, and RAVC may actually be distributed into 3, RAV1, 2 and 3. Okay. Then there is a palm interacting domain, and we know about this palm sequence now, and these are overall the domains of Cas9 protein, which we have already discussed, and in 3-dimensional space they are arranged in this spatial distribution, and this is the linear genetic gene sequence arrangement, but when it is expressed into a protein, it will fall in 3-dimensional space, and then you find the various domains in 3-dimensional space in some structure simplified like this. Okay. Now, let us look into the structure of a Cas9, which we have seen here when it is complex to a single guide RNA, okay, and a target DNA. Each protein domain is colored here according to the domain organization diagram below these structures. The REC1 domain is the largest, and it is responsible for binding the guide RNA. The role of the REC2 domain is not understood currently. The arginine rich bridge helix is crucial for initiating cleavage activity upon binding of target DNA. The palm interacting domain confers palm specificity and is therefore responsible for initiating binding to target DNA. The HNH and RAVC domains are nucleus domains that cut the single stranded DNA. So, with these we complete the understanding of the structure of these very important protein. Thank you for your patient hearing. In the next lectures, we look into the various stages by which the CRISPR-Cas immunity system matures. Thank you.